Welcome, everyone. Today, we're diving into something that sounds like science fiction, but is happening in labs right now. We're talking about a completely new way to build super plants and how it could protect our food from its oldest, most stubborn enemies.
You know, it's easy to forget, but every crop out there is in a constant war against disease. We've been fighting that war for centuries. But what if we could stop fighting battles and just win the war? What if we could give our food a permanent suit of armor? Well, that's exactly the mission we're going to explore. You see, for as long as we've been farmers, we have been locked in this relentless arms race with plant diseases. We breed a stronger plant, and then, sure enough, the disease evolves right back to defeat it. The problem is, our old methods for creating these stronger plants, they're starting to lose the battle.
So just think about the traditional way of doing things. It's powerful, for sure, but man is it slow. We are talking decades just to develop one new disease-resistant variety. And it's all kind of based on guesswork, right? You have to plant the crops, wait for them to grow up, hope they get exposed to the right disease, and then you just watch and see if they survive. It's not exactly efficient. There's got to be a better way.
And, well, there is. Instead of just waiting and watching, scientists are now decoding the playbook hidden inside the plant's DNA. They're literally learning how to see a plant's future. And this right here is where everything starts to change. So let's get into it. The new approach is called marker-assisted selection, or MAS for short. The idea is brilliant. Scientists use these things called DNA markers. Think of them like little genetic flags or signposts to find the exact genes they want. And they can find these genes, like the ones for disease resistance, long before you'd ever see any physical sign of them. It's basically like having a genetic scanner for every single plant.
And the process itself, it's incredibly efficient. So first, scientists figure out which DNA marker is always hanging out right next to the resistance gene they're looking for. Then, they can take a tiny little leaf sample from a seedling, just a baby plant, and scan its DNA for that marker in the lab. And boom, they know instantly if it has the gene. From there, it's simple. They only plant and grow the seedlings that tested positive. No more guesswork, no more waiting whole seasons.
And look, this isn't just some theory. It's a real technology with a pretty sophisticated toolkit. Scientists have all these different types of markers they can use, things like SSRs, SNPs, and this really efficient one called CASP. Now, you don't have to remember the names, just know that each one is a different tool for the same job, making the breeding process way faster and way more precise.
Okay, so now we have this amazing, super precise tool that lets us find the right genes. So, what's the master plan? Well, it's not about finding just one hero gene anymore. The big idea now is to build the ultimate plant by assembling an entire team of them. So let me ask you this. When you're up against an enemy that is constantly evolving and trying to get past your defenses, what's better than one hero gene?
A whole team, of course. And that's the crucial idea here. Instead of relying on a single line of defense that a disease can eventually figure out how to beat, scientists are now building plants with multiple, layered defenses, all working together.
This whole strategy has a name. It's called gene pyramiding. It's pretty much what it sounds like. You take 2, 3, sometimes even more of these valuable resistance genes, often from different parent plants, and you stack them all together into one single elite plant. The goal here isn't just to be strong, but to have a resistance that's broad and, most importantly, durable, something that will last. And the strategic advantage of this is it's just brilliant. First, you get durable resistance. It's just so much harder for a disease to evolve a way to beat 3 or 4 different defenses at the same time. You also get broad-spectrum resistance, so the plant can fight off different versions of a disease. It all adds up to a much smarter, more sustainable way to keep our plants healthy, and maybe use fewer pesticides in the process.
Now, this all sounds great on paper, but you're probably wondering, is it actually working in the real world? And the answer is a big, resounding yes. So let's look at some incredible recent wins where this science has created some seriously supercharged crops.
Okay, first up, wheat. Researchers took on 2 really nasty diseases that often show up together. One is called Fusarium head blight. It actually contaminates the grain itself, and the other is powdery mildew, which just sucks the life out of the plant. The mission was to build one wheat plant that could defeat them both at the same time.
So to do this, they developed a brand new system called multi-CASP. And this was the real game changer. It let them scan for 2 completely different genes in one single test, which made things so much more efficient. And the results? They were crystal clear. The plants that had both genes pyramided inside them were way more resistant to both diseases.
And hey, you don't have to take my word for it. The researchers who did this study said it best.
They concluded that this new system significantly streamlines marker-assisted pyramiding and enhances breeding efficiency. In other words, they figured out how to build these super plants a whole lot faster.
All right, but what if you could take it even further? Next up, let's talk about rice. Because here, scientists went even bigger. They managed to pyramid 5 different resistance genes into a single new plant. They stacked genes that could protect against 2 of the most destructive rice diseases on the entire planet. 5 genes. And here it is. Here's the proof in the pudding. This new, pyramided rice wasn't just tougher, it was actually more productive. I mean, look at that. It yielded almost a full ton more per hectare than its very good parent plant. And that's the ultimate goal, right? Not just to save the crop, but to actually grow more food.
So, this technology is, well, it's undeniably powerful. But like any major scientific leap, it's really important to look at the whole picture. So let's talk about both the incredible promise and some of the real-world practical challenges.
Okay, the advantages are pretty obvious by now, right? You get incredible precision, a huge boost in speed, and the potential for resistance that really lasts. But it's not a magic wand. This stuff is expensive. It requires serious lab infrastructure. It also needs highly trained scientists to do it. And there are still tricky technical hurdles, like making sure you don't accidentally bring a bad gene along for the ride with a good one. It's a major investment.
Which really brings us to the big final question. We now have this incredible power to design and build plants with a speed and precision that was pretty much unimaginable just a generation ago. So, knowing that this is possible, what should the future of our food look like? That's a huge question, and it's something for all of us to think about. Thanks for tuning in.